Hi, I'm Stuart from the Norfolk Honey Company and welcome to another session of Microscopy for Beekeepers. These sessions are sponsored by Brunel Microscopes, who very kindly supplied me with various pieces of equipment that I can use. Today particularly we're going to be using the Brunel Microscope SP50, which is a compound or high-powered microscope, because today we're going to be checking for Nosema. And this follows on from the previous video where we were looking at Acarine, and we're going to take a sample of bees and we're going to test for both Nosema apis and Nosema ceranae. But first we're going to take a look at exactly what Nosema is and how it spreads through the colony and how it spreads between colonies, and then we'll get some samples out and we'll take a look at the samples under a microscope. I've got some that I did previously which were heavily infected with Nosema, so if we get lucky for me and my samples don't have any Nosema spores, then I have some slides that I can show you from a previous testing. If this is your first time watching one of my videos, you're very welcome. Please do hit the subscribe button. We've got lots of other videos in the back catalogue, not only in the Microscopy series but also in our Getting Started series for beginner beekeepers, and also in our Beekeeping Basics series for more general beekeeping tips and techniques. And you're very welcome to drop any questions or comments down in the comments section below, and I promise to answer all that I receive. And any information that you need to catch up on, I'll pop links down into the description box so that they're later as you go through the video. So Nosema is an infection of the gut of the honeybee, and it can take the form of a really nasty dysentery type infection where the bees become very lethargic. The Nosema actually feeds off the cells of the inside of the gut, and this can cause the bees to become incredibly tired, lethargic, and not able to fly out any real distance. And so again, you'll find bees that are maybe crawling. If you open the hive up, you'll find that the bees have been maybe unable to go out to have a toilet break. You'll find that they will have defecated on the top of the brood bars and throughout the hive, and that actually happens to be one of the ways in which the Nosema spores are spread throughout the colony. So what we need to do is try and identify what the problem is, if it is Nosema, and then we can take some action to try and reduce the effect and try and remove the Nosema infection from the colony entirely. That's become quite difficult here in the UK because we have no authorised treatments for Nosema, and so we have to rely quite heavily on comb exchange, and that's something that hopefully you'll be familiar with, but it's basically where we replace the combs on a regular basis and therefore remove hopefully millions of spores from the colony so that it then drops down to a manageable level. So when a healthy honeybee takes up a Nosema spore, it could be because it's cleaning up some cells, it could be that it's just walking around inside the hive and it's feeding and it's picking up spores because the spores are already inside the colony. And once spores pass through the mouth parts of the bee, and the spores eventually find their way through into the ventriculus of the honeybee, and here they will attach themselves to an internal cell, and then they use an interesting technique whereby they use a part of the spore called the polar filament, which everts very much like if you were to take some rubber gloves, turn them inside out and then turn them back out again and squeeze air through into the fingers. They evert in a manner similar to the way that the glove would evert the finger back into its normal state. So as I was saying, if you imagine that, let's get this right, get some air into here. So if you imagine that this is the Nosema spore, and when it penetrates into the cell, it pushes the polar filament into the cell, very much like the glove on this on these washing up gloves. So I'll just draw a very brief basic outline of what a Nosema spore looks like and how this filament is placed inside the Nosema spore. When we look at it through the microscope, you can't actually see any definition in terms of the polar filament, but what you can sometimes see are the two nucleus elements within the cell. So they look very similar to a small grain of rice, and that's kind of what we're looking for when we look through the microscope. And I'll just put up a drawing and then I can show you the various parts of it that we wouldn't be able to see under the microscope but that have previously been described. Okay, so this is basically what we see through the microscope, and then we have inside here we then have another cavity, and then this is the polar filament which sits inside, and then it kind of spirals inside like so, and somewhere in here we'll have the two nuclei. Okay, and so what happens is when it makes contact with the cell, this polar filament is then pushed through and into the cell, and that's how it then connects with its host. So that's a very rough drawing, but hopefully that will give you an idea as to how the Nosema spore actually gets into the honeybee cell. Okay, so that's my very rough drawing. What we'll do now is we'll get on with the actual microscopy section. So we've been out and collected some honeybees. We need a sample of 30 honeybees, and if by any chance you're unable to carry out this process yourself, then please do head over to my website. I'll put the details and a link down below in the description, and we can help you with a check on Nosema. One of the things you might spot is on the outside of the hive you might find that there's a lot of spotting from bee poop as the bees have come out and tried to fly. They can't fly very well, and so they just literally go to the toilet outside the hive. So if you've got lots of spotting on top of the hive, then that could also be an indicator that you may have Nosema. So it's worth getting it checked out, and if you go over to the website you'll find some details there. So we've got our sample of 30 bees, and what we do is we crush the bees up in a pestle and mortar to grind up the ventriculus, which is in the abdomen, and that then, if there is Nosema present, that will expose the Nosema into the pestle and mortar. We add a small amount of water, and in grinding it up we can then take a small sample from the liquid that we have, place that onto a microscope slide. We add a stain called nigrosin, which is a grey black coloured stain, and that stains the spores quite clearly, and we're then able to identify them under the microscope. We use the microscope at 400 times magnification, and the Nosema spores show up quite clearly. So we'll get the camera down and we'll show you what we do. Okay, so we've got our sample of bees with the hive identifier and the date that we took the sample. Pestle and mortar, and we empty the bees out. The bees were killed using ethyl acetate, which is a very quick way of killing the bees. So here we've got our sample of bees in the pestle and mortar, and the next stage is to add a small amount of water, and then we have to grind those bees up. So we just add a few mils of water just to allow us to loosen up the sample. And for those of a squeamish disposition, I would look away now, because we just have to crush all of these bees to break open the ventriculus, so that any spores that are in there will come out into the liquid that we're producing. And then we just take some of the liquid away from the bees. You can now see that we've got a sample of liquid here, and we're going to transfer a small drop of that onto a microscope slide. So the way we take the sample is purely taking our pestle and just dipping it into the liquid and transferring a small drop onto the microscope slide. And that's our sample. And we're now going to add some nigrosin stain to this sample. So this is the nigrosin stain. As you can see, it comes from Brunel microscopes. This I actually purchased some years ago, and you use very small quantities, so it's probably going to last a lifetime really, but it's available from Brunel microscopes. And all we do is take the top off and use a very small drop using our dropper, and just place it into the sample and give it a small swirl, remembering to place the lid back onto the stain so that you don't tip it over and spill it everywhere. When you've finished with the dropper, remember to wash it, because you will have touched the sample and that will contaminate the end of the dropper, and you don't want to then transfer that back into your stain. So always make sure that you wash the dropper after you've used it, and certainly in between samples. And then the next thing to do is to just take the cover glass or the cover slip in the tweezers, and then we just gently position it on top of the sample and allow it to drop down onto the sample. We'll just square it off, and then we've got the sample ready to go under the microscope. Okay, so this is at 40 times magnification. We're just going to focus it in, and there won't be a great deal to see. Here you can see just really general debris from the sample that we've just taken. So what we need to do is to go up in magnification. So this is now 100 times magnification. And you can see various bits of debris. There'll be bee body parts in there, there'll be some fine hairs, we may see some tracheal tubing and general parts of bees. That actually looks like it might be tracheal tubing there in the centre of the picture. But in order to see the Nosema, we need to go to 400 times magnification. So we'll switch to that now. And here we're actually on that piece of tracheal tubing that I just mentioned, and you can see the structure of the tubing quite nicely there. But that's not what we're looking for today, so we'll just move across and we'll come to the edge of the slide, and then what we have to do is to go through a process of very specific searching, because we want to check the entire slide. So what I'll do is go through the process of using a grid pattern. So I'll find one corner of the slide, and then we'll move from that corner and focus everything up, and then we'll work our way through the entire sample, and then I can say whether or not there's any Nosema spores in it. So I'm going to check this sample. I'll put it on fast forward shortly so that you don't fall asleep, and then we'll catch up again when I've finished the entire sample. And hopefully we won't find much, but I anticipate that there'll probably be maybe a few spores here that we'll be able to see. So here, right in the middle of the image, you can see that round object looks like a short, squat grain of rice, and you might just be able to see at the top that there looks to be something coming out of the top of it, and although I can't say for certain, it looks as if that might well be the polar filament that has maybe ejected from this particular Nosema spore. But that's a Nosema spore. It's Nosema apis. And there, as I said earlier, there are two types of Nosema, Nosema apis and Nosema ceranae. And the Nosema ceranae tends to be a more elongated, slightly banana-shaped spore. But this one in the middle of the picture is definitely Nosema apis. And so this is the first spore that I've found. So I think it's safe to say that this particular colony has a very light infection, and I'm going to just go through the rest of the slide to see if I can find any more. But that's quite an interesting image, as I say, with that what appears to be the polar filament coming out of the top of the spore. So I've been through the entire slide, and it appears that I've only found one Nosema spore, which then starts to make you think that maybe it's not a Nosema spore at all, but it certainly has the right shape and size to it. So either I've become very good at spotting a single Nosema spore, or maybe that's not a Nosema spore at all. But there's lots of other interesting things to look at within the slide itself, and it's always good to know that a colony of bees isn't infected with Nosema. So what I'll do is I'll now pop up a slide to show you exactly what Nosema looks like when you have a very heavily infected colony, and you'll be able to see exactly what the spores look like, and we can perhaps compare what these spores look like to the spore that we saw earlier in this slide. So whilst it's disappointing not to be able to show you some live Nosema spores in the slide that we've just had a look at, it's also quite comforting to know that the colony is in good health and surviving the winter quite well. But let's take a look at the Nosema slide that I had from a previous colony, and this was taken a few years ago, and you'll be able to see exactly how many spores you can get in a heavily infected colony. So this is what a colony of heavily infected bees look like, and you can see that there are hundreds of Nosema spores in this image. And interestingly, we've got both Nosema apis and Nosema ceranae here, and I can show you those. What I'll do is I'll put a label on here for Nosema apis, and you can see that the Nosema ceranae has a more elongated shape to it and has a slight curve, a banana shape. And I was once told that to remember the difference, Nosema ceranae is a banana, so it's cerana banana, and that's just stuck with me. So if you want to have a way of remembering which shape is which, then if you think cerana banana, and that will give you a good memory jogger, and you'll know that the more rounded version is Nosema apis. And if we compare the two side by side, here's the one from today, and you can see the single Nosema spore in the middle of the picture, and then looking at the heavy infection, you can see that this particular colony needed a lot of work. We had to change out all of the frames and really tried to help the colony back to full health. Okay, so there we are. There's the test for Nosema, and I'm going to carry on now with some more tests on other colonies. Remember to make a note of the test results that you have. If you've got this far and you're unable to do your own testing, please remember that we can do that for you, so please take a look at our website, and I hope that some of you are now able to take to the microscope with a bit more confidence and know exactly what you're looking for with Nosema. Once again, my thanks to Brunel Microscopes for sponsoring this series of videos, and don't forget to pop over to their website and have a look at all of the great gear that they've got, and we'll catch up again with you on Sunday for the beekeeping basics videos, on Wednesday for the getting started, and again next week for more microscopy in beekeeping. Thanks for watching.